Back in the 1950s, a lot of time and effort was going into genetic research. Early in the decade, Rosalind Franklin and Maurice Wilkins had been using X-ray diffraction photos to figure out the structure of DNA. And in 1953, the famous Watson and Crick carried on from this and proposed the double helical structure of DNA. They also proposed DNA replicated in what we now call a semi-conservative fashion. Now, when they were figuring this out, there were three main theories of replication that everyone considered the most reasonable. These are now known as the dispersive model, conservative model, and semi-conservative model. The dispersive model was the idea that the parent DNA strands were able to replicate and separate at the same time by forming a bubble-like structure in the DNA. The conservative model saw a new daughter helix being created without using any molecules from the parent helix in its final structure. The semi-conservative model involved the two parent strands breaking apart and then each being used to create a new helix that contained both a parental strand and a new complementary daughter strand. For Meselson and Stahl, they wanted to finally figure out which model was actually being used by cells generally, but in particular, cells from the bacteria Escherichia coli. To do this, they did a whole bunch of stuff, but started off with what is called radioisotopic labeling. For their experiment, labeling involved saturating E. coli cells in a heavy nitrogen isotope, N15, that could be integrated into the DNA structure of the first generation cells. The cells were then transferred to a growth medium containing an N14 isotope. Samples were taken at various points in the culture's growth, processed, and spun down in a centrifuge to separate strands of DNA based on weight. They then used a specialized machine to create UV absorption photographs, showing the separated DNA samples in density bands. Each band on the photos was a concentrated section of DNA fragments with similar densities. What they got was something like this. Bit weird to look at, I know, but I'll walk you through it. For this first sample, taken from the first generation of cells, there was one big dark line indicating a really high concentration of DNA with that particular density. By the second generation, the DNA band had shifted further to the left, meaning the cell's DNA helices had gotten a little lighter. They could see the cells were replicating their DNA, but now had to incorporate the lighter N14 isotope. After a few more generations, they found that the band had shifted even further to the lighter left side. By this point, they had ruled out the dispersive model, given there were neat DNA bands and not a bunch of random low concentration bands. They had also started to rule out the conservative model. Okay, so, if only the parental DNA contained N15, only DNA with some N15 and also some N14 could be an intermediate weight, right? They supported this conclusion by mixing samples from the first and final cell generations and taking more UV photos. From this, they made this last little picture. This picture is quite special because it shows three separate bands. One on the right, representing fully N15 labeled parental DNA, a band on the left of unlabeled DNA, and an intermediate half-labeled band situated perfectly in between the other two. This led Meselson and Stahl to conclude the paper with this. We find that the nitrogen of a DNA molecule is divided equally between two physically continuous subunits that, following duplication, each daughter molecule receives one of these, and that the subunits are conserved through many duplications. This diagram was the culmination of their findings and supported the semi-conservative model of DNA replication as the most plausible explanation. Research continued with a range of twists and progressions. Another group of scientists in 1959 performed some experiments and even concluded DNA was quadruple-stranded with double-stranded subunits. But people were quick to disprove them, and ultimately the Watson and Crick model prevailed. John Cairns, in a 1962 paper, found that after processing, E. coli DNA untangled itself to reveal two long, unbroken threads, which further supported the idea that DNA was double-stranded. Not only did it provide arguably some of the most concrete evidence for the semi-conservative model, but the Meselson-Stahl experiment has provided the basis for our current understanding of DNA replication mechanisms, evolutionary genetics, inheritance patterns, and a range of other scientific discoveries. Today, we know that DNA splits apart, allowing a whole range of enzymes to rush in, unwind the helix, bind free-floating nucleotide bases to the parent DNA, and seal it back up to create two whole helices. The implications of all this knowledge have been massive for medicine and biotechnology. It has allowed for improved diagnoses of genetic disorders and enhanced treatments that greatly improve the length and quality of people's lives. This has been the culmination of years of work that will continue well into the future, following epigenetics and the complicated lives of other molecules such as RNA, but no doubt, Meselson and Stahl will remain a cornerstone of this research.